Okay, hello to all of you. Let's discuss chapter number 12. Now, chapter number 12 is a very, very long chapter and it has been divided into two parts. So, I usually I have distributed it into 12A and 12B. So, in the 12A, which is basically the endomembrane system. So, in 12A, we are going to focus on the endomembrane system and we are going to go over the components of endomembrane system. And 12B, I'm going to focus on the endo and the exocytosis, and we are going to talk in much detail about the different proteins which are involved in endo and the exocytosis. So, let's first of all, so this is 12A. So in this, I'm going to talk to you about the endomembrane system. So I hope you know what endomembrane system is because you have already studied in your previous classes. So basically, you know that. And I think when we were having our face-to-face classes, that also I've discussed with you that our cell is nothing but a membrane. It is surrounded by a plasma membrane. And then we have nucleus. Nucleus is surrounded by a nuclear membrane or nuclear envelope, and this nuclear membrane, it actually extends into the cytoplasm and it forms the endoplasmic reticulum. And then we have Golgi apparatus. This is also basically made of membranes. Then we have lysosomes. So the basic components of endomembrane system, we are going to focus. Basically, the endomembrane system involves your plasma membrane, nuclear envelope, all the membranes, then Golgi and lysosome. But in this chapter, we are going to focus on endoplasmic reticulum, Golgi apparatus, and the lysosomes. So basically, you know that I'm going to discuss very shortly that endoplasmic reticulum, is responsible for making lipids and the proteins. And these lipids and proteins, they move from ER to Golgi, and they get furthermore modified and they get glycosylated. So we are going to talk in much detail. So endoplasmic reticulum, which is basically the extension of the nuclear envelope, Golgi apparatus, these are basically membranous sacs, and these are the lysosomes. So lysosomes are basically formed in the cells. So basically anything which is entering into the cell, it enters as the endocytic vesicles, and the Golgi apparatus, so the enzymes, so lysosomes are basically the garbage disposal system, and they have some hydrolytic enzymes in them, and those enzymes are actually made in the Golgi. And those enzymes, they are secreted out, or they are released from the Golgi in the form of the transport vesicles. So these transport vesicles, they have these enzymes, the lysosomal enzymes, and these transport vesicles, they actually fuse with the endocytic vesicles. So these endocytic vesicles, which are coming from the outside of the cell, they fuse with these transport vesicles having the lysosomal enzymes, and the early endosomes are formed. And these early endosomes then further get more developed. Some changes happen in them, which we'll be talking shortly. And then they become the late endosomes, and then the late endosomes finally mature into the lysosome. So basically, this is the endomembrane system. So let's talk about endoplasmic reticulum. So some of the things which you have been studying in the previous classes, I'm not going to go over them. I'm going to go over some of the things which will be new to you. So you know that ER is a continuous network of flattened sacs, tubules, and vesicles. So they are basically two kinds of endoplasmic reticulum: rough endoplasmic reticulum and smooth endoplasmic reticulum. So, so ER is involved in the production of synthesis of proteins. Rough ER is basically involved in the production of proteins, and smooth ER is involved in lipid synthesis. And your rough endoplasmic reticulum, so the ribosomes, so it's called rough endoplasmic reticulum because of the presence of the ribosomes on the cytoplasmic side of the membrane. And smooth ER, it lacks the ribosomes. And this is some morphology. You can go over them here. So these are the electron micrographs. You can see these are the rough ER. This is smooth ER. So rough ER has a rough texture because of the ribosomes which are embedded on these. Because of the ribosomes, they are embedded on the ER. And smooth ER, smooth endoplasmic reticulum, it lacks the ribosome. So that's why it has a smooth texture. The cells which are involved in the synthesis of secretory proteins, they have a lot of rough ER in them, and the cells which are involved in the production of steroid hormones, because steroid hormones lipids, they have a lot of smooth ER in them. Okay, so rough ER has ribosomes on them, and you know that ribosomes are protein factories. So that is why rough ER is involved in the production of proteins. So the ribosomes on the cytoplasmic side of the rough ER synthesizes both the membrane bound and soluble proteins for the endoplasmic reticulum. So the proteins, they can be secretory proteins, which means the proteins which will be secreted outside can be they can be membrane bound. So the ribosomes which are present on the cytoplasmic side of the rough ER, they can synthesize both membrane bound as well as soluble proteins. So we're going to discuss them shortly. Newly synthesized proteins are inserted into the ER system through a pore complex as they are synthesized. So newly synthesized proteins. So let me show you here. So this is the ribosome, this is the mRNA, and this is the growing polypeptide chain. So if you remember the process of translation, this mRNA, it binds on the ribosome, and ribosome actually moves on the mRNA, and the polypeptide chain is made. Now we are talking about the ribosomes which are embedded on the ER. So newly synthesized proteins are inserted into the endomembrane system through pore complex. So we are not talking about the free ribosomes, we are talking about the bound ribosomes because they are present on the endoplasmic reticulum. So these proteins, while they are being made, they are inserted into the endomembrane system through pore complex as they are synthesized. So this process is called co-translation. Okay, co-translational translocation. So this is the ribosome, okay, and this is the mRNA, and this is the polypeptide chain which is being made. So you can see that this is the endoplasmic reticulum, and this is the ribosome. This is the ribosome which is this is the ER. This is the ribosome which is embedded on the ER, so it's a bound ribosome. And this is the polypeptide chain which is being made. This is the mRNA. So while the polypeptide chain is being made, it will be transferred into the endomembrane system. So it is, while it is being translated, while the polypeptide chain is being translated, it will be translocated into the endomembrane system. This process is called co-translational translocation. Okay, so remember that ER has a lumen inside. Okay, so you can go back here and you can check the structure of the ER. So these are called the ER cisternes, and inside this is the lumen, and you can see here these are the ribosomes which are present outside the ER. So the proteins, they go inside the ER lumen while they are getting translated. So that's what I'm explaining. Now as I just said, there are two kinds of proteins, and they can be secretory proteins and they can be proteins which are actually embedded in the membrane. So let's see how they get into the ER system, endomembrane system. So this is the case of the secretory protein. So this is the ribosome, this is the mRNA, and this is the polypeptide. So the signal recognition particle SRP binds to the signal sequence in the amino terminal end of the growing polypeptide chain and halts the translation. So this is the polypeptide chain which is being made by the ribosome, and here is a signal sequence in the polypeptide chain. So SRP, which is a signal recognition particle, it goes and binds here, and it halts up for a moment, it halts up the process of translation. And then here the SRP, which is the signal recognition particle, it binds to the SRP receptor. Here is a receptor. So this particle binds to this receptor on the ER membrane. Okay, so this is the way it brings this whole complex here. So SRP binds to the signal sequence in the polypeptide chain, and then SRP binds to the receptor. So you can see here the SRP receptor brings the ribosome to a transmembrane channel. So here is a transmembrane channel. The SRP disassociates, so this particle, it disassociates. Protein synthesis resumes. So the protein synthesis resumes, which means the translation process resumes again, and growing polypeptide chain is threaded through the channel. So this is the channel, so through which this peptide is going to be going inside. So this is ER lumen. This part is ER lumen. So the protein ends up in the lumen of the ER, where it may remain, be transported to the lumen of another organelle, or be secreted out of the cell. So this is the way how this protein gets into the lumen of ER. Now this protein is in the lumen of ER. So now as the endomembrane system is all acted, so this protein can go to Golgi and it can become part of Golgi, or this protein can also go outside the cell by the formation of vesicles. So this protein which is in the lumen, it can be either a secretory protein, it can go out of the cell, or it can become part of some other organ like Golgi. Now this is the case of the proteins which are actually embedded in the membrane. So let's see how they get embedded in the membrane. So proteins with signal anchor sequence. So the proteins which get embedded in the ER membrane, they have a special sequence in them that's called signal anchor sequence. So the proteins with signal anchor sequence are threaded through a channel in the ER membrane. So they get into the ER through this channel. They get into the channel. So proteins with signal anchor sequence are threaded through the channel in the ER membrane until the signal anchor sequence is encountered. So the protein will enter through this channel, and it will keep on entering till it encounters the signal anchor sequence. The ER channel releases the protein into the membrane, and then at that point this protein is going to be released into the membrane, and this protein will become part of the membrane, which means this protein will be embedded in the membrane. So this is the way how membrane proteins, they get embedded into the ER membrane. Okay, so some of the important functions of rough ER. First of all, its main function is the formation of the, so you have just seen that rough ER has ribosomes in it, so it makes the proteins. And also other things which take place on the rough ER is the initial steps of addition of carbohydrates to glycoproteins. So we are going to see very shortly in detail how the glycosylation of the proteins takes place in the ER. Folding of polypeptides, it also takes place in the ER, folding of polypeptide chains. Recognition and removal of the misfolded proteins. Assembly of the multimeric proteins. So these are some of the important things going on in the rough ER. And smooth ER is also doing a lot of functions. One of the important function of smooth ER is drug detoxification. So there are some enzymes in the smooth ER, and because of which some of the drugs gets detoxified. And how do they get detoxified? By a process called hydroxylation. So these drugs, there are some enzymes in the smooth ER. There are some enzymes in the smooth ER, and because of which the hydroxyl groups are attached to these drugs, and these drugs become soluble, and they can be excreted outside the body. Carbohydrate metabolism is also a function of the smooth ER. There is an important enzyme in smooth ER called glucose 6-phosphatase, and this enzyme is involved in the release of glucose from glycogen. So you know glycogen is a storage form of polysaccharide in animals. It's stored in in the liver and the muscles. So with the help of the enzyme glycogen phosphorylase, first of all, from glycogen glucose 1-phosphate is made, and then with the help of another enzyme called phosphoglucomutase, glucose 1-phosphate is converted into glucose 6-phosphate, and this glucose 6-phosphate is then converted to glucose with the help of the enzyme glucose 6-phosphatase, which is there in the smooth ER, So then this glucose can be through the glucose transporters, it can go to the blood. Also, the smooth ER is involved in the storage of calcium ions. So there are some calcium binding proteins in the smooth ER, and because of that, smooth ER can store calcium ions. The sarcoplasmic reticulum of muscle cells is an example of smooth ER that specializes in the calcium storage. The steroid biosynthesis, as I said, smooth ER is a site of cholesterol and steroid hormone synthesis, and all these are lipids. And also, ER plays a central role in the biosynthesis of membrane. So basically, the fatty acids are synthesized in the cytoplasm and they're incorporated into the ER membrane on the cytoplasmic side. So you can see here, this is the, you know that in the membrane, so we're talking about the ER membrane right now. It has two, it's a bilayer. So this is the cytoplasmic side and this is the luminal side. So the lipids, they're made in cytoplasm and they enter into the cytoplasmic side. So the fatty acids, they are made in the cytoplasm and they enter into the cytoplasmic side. And then with the help of some proteins which are called flipases, or we can also call phospholipid translocators, these fatty acids can move from cytoplasmic side to the luminal side. And this is the way how fatty acids, they become part of the ER membrane. And as you know that the whole ER endomembrane system is all connected. So this this membrane can go and become part of another organelles in the endomembrane system. And similarly, the phospholipids which are present in the ER membrane, the phospholipids from the ER, they can be transported to the membranes of mitochondria or chloroplast with the help of the proteins called phospholipid exchange proteins. So this basically, you can see here, this is a phospholipid here in the ER membrane. With the help of this protein, they can be taken to the cytoplasm, and then from the cytoplasm, these phospholipids can be transported to the membrane of mitochondria or chloroplast. And proteins called phospholipid exchange proteins. Okay, so that's all about the ER. Now the next organelle is Golgi apparatus. So Golgi apparatus, as you have heard in your earlier classes also, it's called the post office of the cell. Why it's called the post office of the cell? Because its function is very much like the post office. Because what's the function of post office? Post office basically collects the letters from different places, and on the basis of the zip code, it sorts the letters and then sent to the destination. So that's also a function of Golgi apparatus. So Golgi apparatus basically receives the proteins and lipids which are made in the ER, and further modification takes place to those proteins and lipids, and then through Golgi, the different proteins and the lipids, they are sent to their destination with the help of the excretory vesicles. The Golgi complex is functionally and physically linked to the ER. So the whole endomembrane system is actually linked. So the proteins and lipids which are made in the ER, they can be transported to Golgi apparatus with the help of the vesicles. So this is all what I said. So Golgi complex plays a central role in membrane and protein trafficking in the eukaryotic cells. So Golgi, you know the structure of Golgi, they are a series of flattened membrane bounded cisternes, and usually there are 3 to 8 cisternes, and that makes a Golgi stack. And if we look here, yeah, if you look here, you can see that these are the cisternes of the Golgi apparatus. These are all membranous. They're all made of membrane. They're membrane. So the cisternes which are towards the ER, that's called the cis Golgi network, or it's called CGN. And the cisternes which are towards the plasma membrane, they're called trans Golgi network. And in between these cisternes, they're called medial cisternes. So the proteins and lipids, they reach from the ER to the Golgi through the help of the vesicles, okay, transition vesicles arriving from ER, and then all the lipids and proteins, once they are all modified, they are transported to the destination with the help of the transport vesicles. Yeah, there are two models. Two models depict the flow of lipids and proteins through the Golgi complex. First model is called stationary cistern model, and the second model is called cisternal maturation model. So according to the stationary model, Each cisterna in the Golgi stack is a stable structure. Each cisterna in the Golgi stack is a stable structure. Transport of material from one cisterna to another. Transport of material from one cisterna to another is mediated by shuttle vesicles. So you can see, and these bud off from one cisterna and fuse with the next cisterna in a cis to trans sequence. So it's like here. So this is a stationary model. So you can, this is the CGN, this is a TGN, these are the medial cisternae. So these, according to this model, these cisternae, they are stable. And cisternae to this cisternae and to this cisternae, it is taking place with the help of these shuttle vesicles. So first of all, a cisternae will form here. So this vesicle which is coming from ER first fuses here, then it gets matured, modified, the protein or the lipid, whatever it is. Then when it has to go from this cisternae to this cisternae, it will form a new vesicle, goes here, then again a vesicle will bud off here and then goes and fuses here. So this is the stationary model. So in the stationary model, transport of material from one cisternae to another is mediated by shuttle vesicles. These bud off from one cisternae and fuse with the next cisternae in cis to trans sequence. According to the cisternal maturation model, the Golgi cisternae are transient compartments. Okay, so which means that according to this model, actually the cisternaes are not stable. They actually themselves keep changing on the basis of on the basis of expression of some enzymes. So these gradually change from CGN through medial cisternae to TGN. So you can see here, this is the these are the cisternae, and how the material is getting transported from this to this side. It is basically because of the change in the cisternae themselves. Another two terms which you need to know: anterograde and retrograde transport. So anterograde transport means the movement of material toward the plasma membrane. So when the transport is happening in this direction, going from Golgi to the plasma membrane, this is called anterograde, and retrograde is coming back from the retrograde is the flow of vesicles from Golgi cisternae back to the ER. So anterograde is moving in this direction, going from Golgi to plasma membrane, and retrograde is coming back from Golgi to the ER. Okay. Yeah. Okay, next we are going to talk about the role of ER and Golgi in protein glycosylation. So proteins, they get glycosylated, which means they get some sugar chains attached to them. Okay, so this process of protein glycosylation, so you remember from your old chapters, some of the proteins are glycoproteins, so which means those sugar chains are attached to them. So how does this protein glycosylation take place? So this process actually starts in the ER. Okay, so both Golgi apparatus and ER both are involved in the process of glycosylation, and we are going to follow the steps. What are the various steps taking place in which specific part of the ER and the Golgi apparatus. So much of the protein processing carried out in the ER and Golgi involves glycosylation. Let's go and see here. And there are two kinds of glycosylations which can take place. It can be N-linked glycosylation. N-linked glycosylation means the addition of oligosaccharide. Oligosaccharide means a short chain of sugar. Involves the addition of an oligosaccharide to the nitrogen atom of the asparagine residue. That's your N-linked glycosylation. O-linked glycosylation, I think we have talked this in previous chapters also. O-linked glycosylation means addition of the oligosaccharide to the oxygen of the hydroxyl group of serine or threonine residues. Now, in the process of glycosylation, all carbohydrate side chains. Okay, so basically, glycosylation process involves the addition of the carbohydrate side chains. So all carbohydrate side chains initially have a common core oligosaccharide. And what does that common core oligosaccharide has? It consists of 2 units of N-acetyl glucosamine. Okay, 2 units. So this pink ring is shown N-acetyl glucosamine. So there are 2 units of N-acetyl glucosamine, 9 mannose units. So these are 9 mannose is a sugar, 9 mannose sugars, and 3 glucose units. So this is this is common core oligosaccharide. Okay, a common core oligosaccharide consists of 2 units of N-acetyl glucosamine, 9 mannose units, 3 glucose units. So how does this process take place? So the initial glycosylation occurs in the ER. So the initial steps of N-glycosylation takes place on the cytoplasmic surface of the ER. Then later steps takes place in the ER lumen. Okay. Yeah. And usually the oligosaccharide is added to the recipient protein as the polypeptide is being synthesized. So this is being shown here. This is your polypeptide chain. This is the amino terminus. This side is the C terminus. As it is being made, as it is being translated, this sugar, this oligosaccharide, this core oligosaccharide is attached to the proteins while it is being translated. So this process is called co-translation glycosylation. Co-translational glycosylation. So this slide actually shows you exactly how this process of glycosylation takes place. So first of all, let's go step by step. Now this is a very important slide to understand. So you can see here, this is the ER membrane. Okay, so this is the ER membrane, this is the ER lumen, and this is the cytosol. Okay, so this is the cytoplasmic side, this is the ER membrane, and this is the ER lumen. So now there is one lipid which is embedded in the ER membrane. It is called dolicol phosphate. Okay, dolicol phosphate is here. It is embedded in the ER membrane. Dolicol phosphate in the ER membrane acts as a carrier of oligosaccharide units for protein glycosylation. Okay, so this is a lipid. Okay, so this is embedded in the ER membrane. Core oligosaccharide synthesis begins in the cytoplasm. So this is the cytoplasm. So it's this process of the core oligosaccharide synthesis. It begins in the cytoplasm as N-acetyl glucosamine and mannose units are added to dolicol phosphate. So first of all, N-acetyl glucosamine is being added, and then mannose units are added. Okay, so once that happens, so once the N-acetyl glucosamine is added and then the mannose units are added, then what happens? So where are they added? They are added to the dolicol phosphate, which is a lipid embedded in the ER membrane. You can see N-acetyl glucosamine is added and these 5 mannose are added. Once this happens, translocation of the oligosaccharide from the cytoplasm to the ER lumen is catalyzed by phospholipid translocator. Now there is another protein here, it's called a flippase. Its function is to flip this oligosaccharide subunit. It's not made yet completely. Its function is to flip it, which means its function is to make this oligosaccharide to move from this side to this side. So it flips and it goes in this direction. It goes into the ER lumen. Earlier it was in the cytoplasm, now it goes to the ER lumen. Okay, and after that, once it goes this side, then there is a because the core oligosaccharide is not completely made yet, so there is the requirement of the addition of 4 more mannose subunits and 3 glucose. So that happens inside the ER lumen. The completion of the core oligosaccharide occurs in the ER lumen as more mannose and glucose units are added. Okay, so that results in the formation of the complete core oligosaccharide unit, and after that this core oligosaccharide unit is transferred to the protein. So transfer of the completed core oligosaccharide to an asparagine residue of the recipient. Right now it is to the lipid. So after that it is transferred to the protein. So it's transferred here, it is shown, to the nitrogen of the asparagine of the recipient protein, and this process is catalyzed by oligosaccharide transferase. It's another enzyme driven process. So this is the glycosylation process starts. It starts in the ER. You can see that how a protein is getting changed, and it is basically the cosaccharide subunit. Final processing involves removal of certain amino acids in ER before transfer of the glycoprotein to Golgi. So this is happening in ER. Okay, this is some part of the glycosylation steps which are taking place in the ER. After that, this protein has to go to the Golgi apparatus. So after this, the final processing take place, and then the protein can go to Golgi operators. Okay, so what is the role of glycosylation? So the function of glycosylation is to help in the proper folding of the proteins. Okay, so there are two proteins in the ER. Okay, they're called calnexin and calreticulin. Their function is basically helping in the proper folding of the proteins, and they bind to the glycoproteins. And then there is another protein called ERP57. It catalyzes the disulfide bond formation. So it also helps in the folding of the protein. So here it shows that how calnexin, so we talked about the two proteins, calnexin and calreticulin, they basically bind to the glycoproteins and they help in the folding, proper folding of the proteins. So basically, calnexin binds to the incompletely folded protein. So if a protein is not completely folded, that protein is not going to form function. So that folding of the proteins, proper folding of the proteins is very important. So this protein calnexin, this is calnexin, it binds to incompletely folded protein. So this calnexin can bind to incompletely folded proteins. So how does this protein recognizes that this protein is incompletely folded? So the proteins which are incompletely folded, they have one terminal glucose on them, and that terminal glucose is actually added on the incompletely folded proteins by the enzyme called glycosylase transferase. Okay, so this glycosyl transferase, it helps in the transfer of glucose to the incomplete folded proteins, and it's a kind of a signal. This terminal glucose is a signal for the recognition of the calnexin. It's a signal that this protein is not completely folded. So this calnexin is going to bind this incompletely folded protein, and then with the help of the another enzyme called glucosidase, this glucose will be removed and this protein will be properly folded, and then when it is properly folded, it can exit from the ER. Okay, but suppose it is not properly folded, then this glycosyl transferase is going to again add glucose to it, and it will be this protein will again have a signal that it's not completely folded, and it will go through this. So basically, glycosylation process helps in the folding of the proteins, proper folding of the proteins. This slide actually shows you the various events which takes place in the ER and the Golgi. So ER, we just talked about biosynthesis of core oligosaccharide. We just saw that how the core oligosaccharide is made in the ER. It starts in the cytoplasmic side of the ER, then some of the steps takes place in the lumen of the ER, processing of core oligosaccharides, removal of misfolded proteins. So all these things take place in the ER we just talked about. After that, these proteins, they move with the help of these vesicles to the CGN, which is the cis Golgi network, and here what happens, attachment of N-acetyl glycosamine. Okay, so the glycosylation process is not complete here in the ER, so it continues in the Golgi. So in the CGN, attachment of N-acetyl galactosamine takes place, then phosphorylation of some lysosomal proteins take place. We will talk about that. And in the medial cisternae, removal of mannose, attachment of N-acetyl glucosamine, addition of galactose, addition of sialic acid. So all these things are part of glycosylation. And in the CGN, attachment of sulfate to tyrosine. So these are some of the events which takes place in the different cisternaes of the Golgi. Yeah, now we are going to talk about that how does a protein knows that whether it needs to go to Golgi, whether it needs to go to ER. So these, or the lipid, where they're supposed to go, how do who is governing them? Okay, so all these proteins and the lipids, they have some tags in them. So each protein contains a specific tag targeting it to a transport vesicle that will take it to the correct location. Okay, so in a protein, the tag can be an amino acid sequence, a hydrophobic domain, oligosaccharide side chain, or some other feature. Okay, And for lipids, they can be one or more phosphate groups attached to positions 3, 4, and 5 on the phosphatidyl in so these tags help them in binding to some receptors, and then these can basically help in getting them um, to um, to the transport vesicles, and they can then take them to the destination. Okay. Okay, so some of the proteins, they stay in the ER because they have some tags in them, um, and they're called retention tags. So a protein is going to stay in the ER because it has a retention tag for staying in the ER, and the retention tag for staying in ER is RXR, where R is arginine, X can be any amino acid, and another R is also arginine. And some proteins which have reached to the Golgi, they can come back to ER because they have some tags in them. They're called retrieval tags. So some proteins return from Golgi to the ER, they contain retrieval tags. And these are some of the retrieval tags. You need to know these tags. So these tags are short C-terminal sequences such as KDEL, or KKXX in mammals, and HDEL in the yeast. So basically KDL is this is basically the amino acid sequence. So this is a tag, this is called the retrieval tag. And how about the proteins which are embedded in the membranes of Golgi? So the Golgi specific proteins, the proteins which are embedded in the Golgi membranes, they're integral membrane proteins with one or more membrane spanning domains. Okay. So the length of the hydrophobic domain may determine into which cisternae each protein is incorporated, because these cisternaes, they have different thickness. Okay, so what is the length of the hydrophobic domain of the protein? The length of the hydrophobic domain of the protein may determine into which cisternae each protein is incorporated. So the thickness of membrane in Golgi increases from CGN to the TGN. So as we go from CGN to medial cisternae to the TGN, the thickness of the membranes, it increases. Thickness of membrane increases as we go from CGN to medial cisternae to the TGN. So the proteins move from compartment to compartment until the membrane thickness exceeds the length of the transmembrane domains. And once that happens, this further blocks the migration and the protein gets embedded there. Okay. So now that's all about the Golgi. Now we are actually moving towards actually not hold on, we are not done with Golgi yet. Now what we are going to talk about, we're going to talk about the lysosomal proteins. Okay, so lysosomes, as we have been studying till now, we have always been studying that lysosomes are membranous sacs. They have hydrolytic enzymes in them, and their garbage disposal system. Anything which is not required by the cell, something which is coming foreign from outside, something entering, something foreign to the cell, something which is entering from outside the cell, or some organelle, some old or the worn out organelles which are not required by the cell, they are all taken care by the lysosomes. So that's true, but the thing is, lysosomes are not like the ready-made organelle sitting in the cell like that. So basically the lysosomes are actually formed in the process. So how they are formed? They are actually formed by the fusion of the endocytic vesicles or the phagocytic vesicles. They are formed by the fusion of the endocytic vesicles or phagocytic vesicles. So when these vesicles, when they fuse with the secretory vesicles which are coming from Golgi, okay, so in the Golgi apparatus, some of the enzymes are made, and those enzymes are the lysosomal enzymes. Okay, so those enzymes are hydrolytic enzymes, but they are not activated immediately. So these enzymes, the lysosomal enzymes, they are made in the Golgi, and they're secreted. These enzymes are secreted by the secretory vesicles, and these secretory vesicles from the Golgi having these enzymes, they fuse with these endocytic vesicles, and with this fusion of the endocytic vesicle with the secretory vesicles coming from Golgi having these enzymes, this fusion forms the early endosomes, and these early endosomes, in these early endosomes, they get converted into late endosomes, and the late endosomes they get converted into lysosomes. So they go from early endosomes to late endosomes to lysosomes, and it is all happening because of the change in the pH in these vesicles. And the pH keeps on becoming low because in these vesicles there are some proton pumps, and these proton pumps keep on sending the protons inside and keeps on making the environment inside these vesicles, these endosomes, it keeps on making it acidic. And these hydrolytic enzymes, which are actually formed in the Golgi, they become activated in a acidic environment. So when these enzymes, they get activated, then there's the formation of lysosomes. So the stuff which I just said is very, very important for your understanding. So right now what I'm going to talk about is the enzymes, how they are formed in the Golgi apparatus. So basically these lysosomal enzymes, they are actually made on the ER. Remember that ER is the one which is having ribosomes. So the proteins are actually made on the ribosomes because ribosomes are the protein factory. So soluble lysosomal enzymes in the ER and early Golgi complex. So they are basically made in the ER and then they come to the Golgi apparatus. So soluble lysosomal enzymes in the ER and early Golgi complex undergo N-glycosylation followed by removal of glucose and mannose units. The mannose residue on the side chains are phosphorylated within the Golgi complex. So this is something very important which takes place in the Golgi apparatus. These mannose residues, they get phosphorylated in the Golgi apparatus and forming an oligosaccharide containing mannose 6-phosphate. And this mannose 6-phosphate, it basically binds to the 6-phosphate receptors, and then this ligand and receptor complex is actually packaged inside some secretory vesicles. So this tag ensures delivery. This tag, so this is a tag, mannose 6-phosphate is a tag. It's a tag for lysosomal enzymes. This tag ensures delivery of lysosomal proteins to the lysosomes. So phosphorylation of mannose is catalyzed by two specific enzymes. The first is phosphotransferase that adds N-acetyl glucosamine 1-phosphate to carbon 6 of mannose. So first of all, N-acetyl glucosamine 1-phosphate is added to carbon number 6 of mannose. Mannose is a sugar, is a monosaccharide in the early Golgi complex, and then the second in a mid-Golgi complex, second enzyme in the mid-Golgi complex removes N-acetyl glucosamine, leaving behind. So this is first. So to the mannose, first of all, this N-acetyl glucosamine phosphate is attached to the carbon number 6 of mannose, and then this, with the help of the second enzyme, this is removed, leaving behind only mannose 6-phosphate. So this mannose 6-phosphate is a tag for lysosomal enzymes. So the tagged lysosomal proteins bind to mannose 6-phosphate receptors. Okay. Then the receptor ligand complex are packaged into transport vesicles and conveyed to endosomes. They're basically these transport vesicles having lysosomal enzymes. They actually go and fuse with the endocytic vesicles and they form the early endosomes. Okay. So in animal cells, lysosomal enzymes are transported from T-gen to organ. Then, so basically, first they form early endosomes, then they form late endosomes, and then eventually they form the lysosomes. So you can see here, this is the lysosomal enzyme is synthesized. So this is the enzyme and this is the carbohydrate part. Okay. Mannose is phosphorylated by the, we just talked about the two enzymes which help in the addition of phosphate group to the mannose. So this is the tag for the lysosomal enzymes. So then this mannose 6-phosphate, it binds. So this is a receptor. This is a receptor for mannose-6-phosphate. So this mannose-6-phosphate binds to this receptor. You can see that. So mannose-6-phosphate binds to the receptor, and the tagged enzymes are packaged into the transport vesicles. Okay, so this is the way. So you can see here, it's shown here actually. So this is the receptor. This is the this is the lysosomal enzymes with mannose-6-phosphate, and it's binding to its receptor, and you can see here it is packaged in a transport vesicle. Okay, and as this vesicle will go through different stages, this vesicle will fuse with the endocytic vesicles, and then it gets converted into early endosome, late endosome. So in the late endosome, what is going to happen? The pH is going to go low. So the low pH in late endosome will cause the dissociation of the complex which is formed between this receptor and this enzyme, and as a result of that, this receptor will be lost. Okay, so low pH in the late endosome causes dissociation of enzyme and the receptor. So it will break their bonding. The receptor and the enzyme will lose their bonding, and this enzyme will be the only one which will be there in the vesicle, and this receptor will go back, this receptor will be recycled. Okay, so this is the way, and then this late endosome will further get converted into a lysosome. Okay, so lysosome, I have been telling you, so this is the way how lysosomal enzymes, they are formed in the lysosomal enzymes are formed in the Golgi complex. So you know what lysosomal enzymes are. I have been telling you a lot of things, so I'm not going to go over these. You can just read these things. So lysosomes, they have an acidic environment. So lysosome will be formed only when the pH is going to drop to this level. And how does it drops to that level? Because of the presence of these proton pumps in the membrane, and they keep on delivering these protons inside, and it brings the pH to be very low. So these things we have all covered. So let me explain you from here actually. So what I have been explaining you, that this is something entering into your cell with the process of endocytosis, or endocytosis is actually of 3 different kinds. It can be pinocytosis, phagocytosis, and receptor mediated endocytosis. So in the next part, I'm going to talk about the receptor mediated endocytosis in much more detail. So this is, you can see this is endocytosis, and this is something solid, as something entering into the cell. Solid is here with the phagocytosis. So these are endocytic vesicles, and this is your Golgi apparatus, and this is all the vesicles, the excretory vesicles or the transport vesicles having all these enzymes are here, but these enzymes are not active yet. So these vesicles actually, they fuse with these endocytic vesicles. So these vesicles fuse with this, and they form early endosome. So this early endosome will change into late endosome, and then it is going to change from late endosome to the lysosome. So the changes which takes place, so the pH of early endosome lumen drops to 6 to 5.5. Okay, so once that happens, these endosomes lose the ability to fuse to the endocytic vesicles, and then they convert into, so they lose the ability to, the late endosome is packaged with material to be digested and with newly synthesized digestive enzymes. Yep. And the last step in the development of lysosome is the activation of the acid hydrolysis, and this occurs because the pH becomes very acidic. It goes from 4 to 5, and this is because of the pumping of protons. I've already explained them. So when the pH goes from 4 to 5, when the pH becomes like this much, then the late endosome is converted into a lysosome, because now all the enzymes which are there in these vesicles, they are going to be activated, because all these are hydrolytic enzymes. They are active in the acidic environment. So these lysosomes, if the lysosomes are containing substances that have originated from outside the cell, they're called heterophagic lysosomes, and these lysosomes can also have the material which is coming from the inside the cell. They're called the autophagic lysosomes. Yes, there are various things inside the cell which cell doesn't need, and they eventually when they combine with these vesicles which are coming from the Golgi, they are called the autophagic lysosomes. Again, coming back to here, I would explain that this slide actually shows everything. As I said, these endocytic vesicles combining with these vesicles which are coming from Golgi, having these enzymes, they convert into early endosome. Early endosome becomes late endosome. It then becomes lysosome. It then converts into lysosome, and in the lysosome, because all these enzymes are active now, they will start the process of digestion. So all the digestion going on, and after the digestion, the nutrients which are released, because so what is happening, all the macromolecules, they're going to be digested, they're going to be converted into smaller subunits. They can be either released to the cell, because the cell can use all these nutrients. So this lysosome can all excrete out these things, the cell can use them, and anything which is not digested in the vesicle stays there, and it is called a residual body. And also the cell can release some of the nutrients by the process of exocytosis. It can also secrete some of the nutrients outside the cell by the process of exocytosis. And there is another possibility that these lysosomes can also secrete some of the hydrolytic enzymes outside the cell by the process of exocytosis. So this is showing exocytosis releasing acid hydrolysis to the extracellular fluid. And this is basically showing an autophagic vacuole which is being formed, which is basically a worn out mitochondria, and it is basically this autophagic vacuole is then combining with these vesicles, and it's also going through all these stages. So as I said, these vesicles can be these material in these lysosomes can be coming from outside the cell, or it can be coming from inside the cell too. So this slide actually shows everything. So this is all what is written all over here. And the last thing I would like to talk about, autophagy and cancer. So this is an important point. So recent work suggests there's a link between autophagy and cancer. So in the mice, there was knocking out of some genes which are required for autophagy. So some of the genes which are responsible for autophagy, if they are knocked out, it was shown that there's a decrease in autophagy. So there was decrease in the autophagy because of the knocking out of some genes which are responsible for autophagy. There was an increase in the breast and the lung tumors. And I think I have posted one video, you can watch that also, which shows a link between autophagy and cancer. And this is already we have discussed. So that's all in this chapter. This is 12A. And if you have any question, please let me know.